Let's learn the chapter Molecular Basis of Inheritance with Mnemonics. Let's begin by looking at the major experiments identifying genetic material. A great way to remember the timeline is the mnemonic Griffith shows, Avery proves, Hershey confirms, Meselson demonstrates. Specifically, Griffith showed that transformation in bacteria occurs where the S strain transforms the R strain. Avery proved that DNA is the actual transforming principle. Hershey confirmed that DNA enters the bacteria, not the protein. And finally, Meselson demonstrated that DNA replicates semi-conservatively. To understand Griffith's work better, we look at the capsule concept using the mnemonic smooth kills, rough lives. This means the smooth or S strain has a capsule and is virulent, causing death, while the rough or R strain has no capsule and is non-virulent. Now, let's move on to the process of prokaryotic DNA replication. The mnemonic here is fork starts, origin fires, polymerase builds, fragments join. This describes how the replication fork forms and the process begins at a single origin called auric. Then DNA polymerase 3 elongates the strand, and finally the Okazaki fragments are sealed by ligase. In contrast, when we look at eukaryotic DNA replication, remember, many origins, S phase works, chromatin slows. This highlights that there are multiple initiation points, the process is restricted to the S phase of the cell cycle, and the nucleosome organization of chromatin slows down the replication. Next, we have the process of transcription in prokaryotes. The mnemonic to recall is 1 polymerase, sigma starts, RNA elongates, rho ends. This tells us that there is only one RNA polymerase involved. The sigma factor is required for initiation. Then the polymerase synthesizes the RNA. And finally, rho-dependent termination occurs alongside intrinsic termination. For transcription in eukaryotes, the process is more complex. The corrected mnemonic is 3 polymerases, introns out, cap on, tail added. This signifies that polymerases 1, 2 and 3 are involved. Splicing removes the introns, a 5' cap is added, and a poly A tail is added at the 3' end. Moving on to protein synthesis, let's look at translation in prokaryotes. Remember, 70S binds, shine guides, F-met starts, stop releases. This means the prokaryotic ribosome is 70S. The Shine-Dalgarno sequence guides the ribosome. The initiator tRNA carries formyl methionine or F-met, and the stop codon triggers the release factor. Comparing this to translation in eukaryotes, the mnemonic is ATS binds, cap scanned, met starts, factors finish. Here, the eukaryotic ribosome is ATS. The small subunit scans from the 5' cap. The initiator tRNA carries methionine, not F-met, and eukaryotic release factors release the peptide at the stop codon. Finally, let's understand the lac operon. The mnemonic is repressor blocks, lactose binds, operator opens, enzymes made. In this process, the I gene produces a repressor. Then, lactose binds, specifically allolactose, which inactivates the repressor. This allows the operator to open so RNA polymerase can transcribe, and consequently, enzymes like beta-galactosidase, permease, and trans-acetylase are produced.
That's it for today. Learn smarter with EduRev. Notes, tests, and practice in one app. Download EduRev now on Play Store and App Store and keep learning every day.